All right, so now to some extent we want to turn the next question to the audience because we now have an educated audience of thousands of these MOOC learners who now have a base knowledge. And what we'd like to do is put to you what we think are the most important questions that need to be resolved because we're all getting old and you guys are potentially have the potential both to impact the next work that's done either by pursuing further education and doing this work yourselves or by advocating for the kinds of research that you'd like to see done. And so the question I put to everybody is, what is the next big thing? What are the most important questions that we need to tackle in infectious disease biology?
Well, I think that over the past couple of years, we have seen evidence of a nice link between microbiota and the ability to be susceptible to a viral infection, your ability to clear a viral infection. The microbiota is linked to so many aspects of just health, homeostasis, and well-being that I think that really is going to have to be an emphasis. We're always looking for changes in nuclear genes for issues of susceptibility. We know that there's an environmental factor, and I think one of those components is the microbiota. And so I think many of us are trained as virologists or bacteriologists, and there's not a lot of cross-fertilization. I think that's going to be more and more important in understanding disease mechanisms.
And there's been a lot of descriptive science that's done, but not a lot of functional or practical science in terms of the ability to engineer. Right? This is a question that's come up in the forum many times, right? How do we get the good microbiota, right? How can we engineer it? Is there a pill we can take that will, right? And it's perhaps a little bit laughable, but the idea of engineering an optimal environment internal environment to protect us is, perhaps science fiction for now, but it certainly has potential down the road.
Yeah.
Thing that I would think about so is plant health. So we think about One Health and needing nutrition and for communities, and 40% of all the plant crop across all the major crops is lost on average each year because of diseases. But unlike animals, you can do pretty much anything you want to the plant. You can kill them with impunity, you can wipe out a crop, you should be able to limit the spread of the diseases. And I would like to see better ways in which we see a better mapping of diseases on a global level. So we can organize communities saying, for example, this area has cassava virus or mosaic virus, and let's just wipe it all out, all the cassava, so the disease runs its course. And we would deal with the vectors of those diseases, which we largely recognize. And so we can remove these and hopefully drive to extinction a lot of these diseases of the plants. Because the difficulty with polio and others are the human community, the elements of humanity, or even animal diseases. We don't find the bat reservoirs, but the plants are right there in the field. So there's no reason why we shouldn't be able to better control plant diseases and effectively remove them, and then recovering all that lost yield, which is quite considerable, especially given the large expected dinner table we'll have in the next 50 years with 9 billion people.
But the point you make about the ability to see these things, I think there's quite a bit of work suggesting that many of the pathogens that we see causing disease in crop plants don't necessarily cause disease in wild reservoirs. And so it may in fact be that it's a very analogous situation to not being able to detect the bat reservoir for Ebola if there are many, many species of plants out there that are viable hosts for some particularly viral pathogens. But they're not expressing any disease, and there are many, many thousands of them, and they're very, very hard to track.
Unlike the bats, the trees, which are the reservoirs of these diseases, don't fly. And so you could actually create these cordons and stop all we know where these insect vectors won't travel. And so you could have a much better agricultural system where, for example, in West Africa where I work, you have rainforest trees right in the middle of cacao or cassava crop, and that's just a crazy idea. Get rid of it, and create this better management practices where you're not having these reservoirs being
That's interesting that you're advocating for more monocultures again, because
I think what we seem to do is keep the very worst of biodiversity in these places, and rather romantically we think a rainforest tree is some good thing to keep around, but either have it all rainforest or have a really good border. So we'll see these in developed agricultural societies like Holland or America. You don't have such spillover effects, and so I think we can actually properly manage it, and that will reduce the pressure on the actual reserves of rainforest, which I obviously like and where I obviously work.
I guess I really agree with Craig to some extent, but with some caution that as we create environments that protect us against what we know now, we mentioned this earlier, that there are environmental things will change, and that which may make us more susceptible later. And so this selection, if you will, for a finer phenotype of resistance to one element may, on the other side, make you much more susceptible down the road. Sure. So one of the things, of course, we do here is deal with dynamics, and this is recognizing that things change in time and space. And so I guess my advocacy here would be for better integration of those dynamics across scales. We deal at molecular scales pretty well, and we deal at sort of large population scales pretty well, but we don't do a good job of, moving amongst those scales.
Mm-hmm. Mm-hmm. And I think so if I can add one other next big thing that I think is important is the question of the other next big thing. What is going to be the next big epidemic? It's a question that constantly gets asked, but it's next to impossible to answer in advance. However, what we can do is perhaps improve our ability to identify potential risks and identify outbreaks when they happen early enough that we can get ahead of them in time to resolve them. So better investment in diagnostics, in basic understanding of parasite biogeography and biodiversity, so we know where are the best places, what are the most efficient places to be allocating resources for surveillance and detection, right? Because as we've seen in many of these cases, right, epidemics tend to take off exponentially, and the sooner you can nip that in the bud, the better your ability to contain the whole thing, right? And so I think better understanding, better targeted approaches to diagnostics and surveillance might help us get ahead of whatever the next big thing is, even though we won't be able to predict it perfectly. Mm-hmm. So those are the set of questions that we hope you guys will spend the next year at least thinking about before Epidemics 3.0 comes back next fall. Thank you.
Bye.